Good morning. I think we'll get started. I'm Dan Mathalon, and I have the pleasure and privilege of introducing our Grand Round session this morning, which is called Connections, Dr. Feinberg and the Pruning Hypothesis. I'm going to start by giving a little bit of background on Dr. Erwin Feinberg, to some of us known as Bob. And since we go back and forth on that one, I'm hedging my bets and keeping both names active here, because I can't help but think of Dr. Feinberg as Bob for all the years I've known him. I'll just say a few words at the beginning, just to give you some sense of Dr. Feinberg's academic background and history. He got his bachelor's in psychology from Brooklyn College, master's in experimental psychology from Swarthmore College, his MD from NYU, and then did residency training at Mass Mental and then at the NIMH St. Elizabeth's. His research career began at the NIMH. He moved to SUNY Downstate, where he rose to the rank of professor. And in 1969, he moved to San Francisco and became the chief of mental health at San Francisco VA, as well as vice chair in the Department of Psychiatry. He had a brief stint at NYU, which I mean to ask him about, but it was a short time before he came back to UCSF and the San Francisco VA, and remained until 1985, at which point he spent 3 years at SUNY Stony Brook, and then moved to UC Davis and the Martinez VA, where he was the director of psychiatric research at UC Davis, and remained as a professor there until he became emeritus recently. He has won several awards, including early on a Career Teacher Award from Downstate Medical Center, Distinguished Scientist Award, from the Sleep Research Society, and School of Medicine Faculty Research Award from UC Davis. He's been the PI of numerous grants, mostly related to sleep research, which he's been doing for almost 60 years now. He has over 150 publications, including articles, reviews, letters, and chapters. And what I want to just highlight today at the beginning are some of Dr. Feinberg's important contributions to schizophrenia research and schizophrenia theory in particular. One that we'll be spending most of our session talking about today, which is the abnormal synaptic pruning hypothesis. But the other I wanted to highlight, for reasons that will become clear momentarily, are his ideas around the possibility that efferents copy corollary discharge mechanisms might be dysfunctional in schizophrenia and account for certain symptoms. When I think about Dr. Feinberg and his contributions to our field, there are two words that really come to mind, and I want to highlight them. The first one is visionary. Now that can sound a little grandiose, but I actually think in this case it really does fit, in the sense that Dr. Feinberg has had a unusual ability to think very integratively and imaginatively about theoretical ideas, what might be going wrong in schizophrenia, well ahead of the development of methods to test a lot of those ideas. They, in some sense, predate and predict future directions in our field in a way that is very unusual. So visionary is certainly one adjective that I would use to describe Dr. Feinberg's contribution. The other is seminal. And here I'm talking about the way that his ideas have influenced others in the field, particularly others who have either adopted those ideas, extended them, in some cases repeated them, and I have to say sometimes not with as much acknowledgement and credit as I would have liked to have seen. Somewhat reminiscent of my experience in the 1970s when I encountered younger people who were talking about the wonderful Van Halen hit, "You Really Got Me," and if any of you are Kinks fans, you know that they didn't write that song. But anyways, back to the matter at hand. Dr. Feinberg's ideas have propagated forward with other theorists and other researchers who have, in various ways, been guided by those ideas in terms of testing research hypotheses. So I just want to mention one very important work published back in 1978, a theoretical paper on the possibility that efference copy and corollary discharge mechanisms might be abnormal in certain types of psychosis or psychotic thinking. The efference copy corollary discharge mechanism is basically a broad set of mechanisms that have been characterized across the animal kingdom that refer to how organisms differentiate the sensory consequences of their own actions from the sensory events that arise from the environment and environmental changes. And the idea was that if those mechanisms aren't working properly, one might start to have experiences like hallucinations or delusions of alien control. And this was very influential in my career development and thinking and in Judy Ford's. And both of us carried this work forward. But I just want to mention that these ideas were picked up by others like Chris Frith, and even very recently in a paper in JAMA Psychiatry, where in this case some younger folks had kind of lost track of the origin of the ideas, but they are indeed going all the way back to Dr. Feinberg's seminal paper. And I just want to tell you a little bit about our work in trying to test this idea. And the way that we did it is we looked at the auditory cortical response to self-generated speech with the idea that the speech areas send a efference copy or virtual copy of that speech plan to the auditory cortex, where it creates a corollary discharge representing the expected consequences of that speech. And if it matches what you actually say, the auditory reafference, then there's a net cancellation and suppression of the auditory cortex. And we see this on the left in healthy controls with the N100 being reduced during speaking relative to when that speech sound is played back. And we see that that is not present in schizophrenia. They do not modulate their auditory cortex when they're speaking relative to playback. A hypothesis that was generated from Dr. Feinberg's theory that this would be abnormal. Now I want to turn to the topic for today. This visionary and seminal paper was an attempt to explain some findings from sleep, as you'll hear probably a bit more about why sleep and a few other areas, what might be going on during adolescent brain maturation that could account for the development of schizophrenia or the onset of schizophrenia or other psychotic disorders in the second decade of life. And this idea, too, I want to emphasize, was proposed before we had a lot of the post-mortem data that I'll just come forward, that tell us that in fact in the brains in schizophrenia there is a reduction of neuropil but not cell loss. There are less branches and connections, fewer dendritic spines. This came after the Feinberg hypothesis was originally proposed. And it also was something that preceded the findings from decades of research on using MRI to study structural brain changes in schizophrenia that documented gray matter loss, including at the first episode of illness. And I remember meeting Dr. Feinberg when I was in Dolph Pfefferbaum's lab at Stanford, because we were doing volumetric MRI studies, and really the synaptic pruning hypothesis was the major hypothesis guiding our thinking about how did the brains get this way in schizophrenia. So just to highlight, Keshavan picked up on this work and integrated it with the MRI findings that came much later. McGlashan and Hoffman also wrote a theoretical paper that was based on the pruning hypothesis originally proposed by Feinberg. And then I want to share one final thing. I'm one of the investigators of a 9-site prospective study of individuals at clinical high risk for psychosis. This is known as the Naples study, the North American Prodrome Longitudinal Study. One of the findings from that study that drove a renewal grant submission was that gray matter was declining in individuals who converted to psychosis. At-risk individuals who subsequently converted lost gray matter over a 1-year period at a faster rate than healthy controls followed over the same period, and relative to non-converters followed over the same period. We formulated a hypothesis and a number of specific aims related to explaining this and trying to pursue it, particularly focusing on overactive synaptic pruning as a possible mechanism during adolescence, which is what these individuals are, they're adolescents. It seemed very plausible that this could be an account of what was going on. And I want to just share a comment from one of the reviewers, the first round, which it did not get funded. It had about a 50th percentile score. What this reviewer said is that the mechanistic model proposed to explain the observed gray matter loss is a resurrection of the Feinberg over-pruning hypothesis advanced 2 decades ago, for which supporting evidence is mixed at best. The mechanistic model that has been proposed to try to explain those findings is mainly a reformulation of models that have been around for a decade or more but remain fruitless. This was in 2013. We did get the Naples grant funded, and we're in year 5 now of that grant. But somewhere within a couple of years of this, this finding came out on the 108 bona fide risk genes for schizophrenia. And then, not too long afterwards, in 2016, this finding came out, which you'll be hearing about today, identifying that peak in the profile of genes as a gene that codes for a protein that is involved in synaptic pruning that is implicated as a risk gene for schizophrenia and implicates deficient pruning. So just a couple of conclusions I want to draw. First of all, I think it's very important for us to track the history of our ideas in psychiatric research, and that is an important part of our educational mission. Secondly, that good theories can precede the development of methods needed to test them. And just because they've been around for a while does not mean they've been disproven or that they lack merit. So, with that background, it's my pleasure to introduce our speakers today, who will tell you a bit about their interest in this story and their work in creating a video to document some of the history related to this. This is a partnership of 2 individuals, Dr. Seth Cohen and Dr. Howard Rosenbaum. Dr. Cohen got his bachelor's from Occidental College in Los Angeles, his MD from Northwestern, and did his psychiatric residency at UCSF. Finishing in '85, he became an assistant professor of psychiatry at the University of Washington in Seattle until '91, at which point he went into private practice in Seattle. And then in 2016, he became a staff psychiatrist at Atascadero State Hospital. He has over 30 papers on schizophrenia, metabolic disturbances associated with antipsychotic medication, and treating behavioral disturbances in individuals with intellectual disabilities. Our second speaker is Dr. Howard Rosenbaum, got his bachelor's from Harvard, MD from the University of Oregon, residency training at St. Mary's Hospital in San Francisco, and he's also a graduate of the San Francisco Psychoanalytic Institute. From 1980 to 1989, he was an assistant clinical professor at UCSF and an attending staff psychiatrist at San Francisco General. From 91 to 2005, he was an assistant clinical professor at Oregon Health Sciences University in Portland. And in the Portland area, he has also served as a consultant, staff psychiatrist, and medical director roles in various hospitals and clinics. And he's been active in private practice now in Beaverton, Oregon. Dr. Rosenbaum specializes in psychopharmacology in his private practice. In addition to his work in psychiatric care, one of his hobbies for the past 30 years has been videography and making films for family, friends, and community nonprofits. And he is currently the president and CEO and most prized employee of Zula Productions, which I have a feeling may be his own company. So, it's a huge company. On that note, I want to ask Drs. Cohen and Rosenbaum to come forward and begin their part of the presentation this morning.
So, hi, I'm Howard Rosenbaum, a psychiatrist in private practice in Beaverton and president and CEO of Zula, that's Zula Productions. I was at San Francisco General back in the early 80s as a young attending, and in I think my second year of attending, I had a young intern, Dr. Seth Cohen. And I've now known him for 35 years. Back then, and together we made this video you're about to see. So back then, the Giants played at Candlestick. It was very cold. They weren't very good, and they often gave us tickets to the game. So the patients would go off to the game with the staff, and one day I said to Seth, you should go along. I think it's a good team. You'll enjoy it. Go along with them. And off he went, and I'll leave it to Seth to tell you the story and a little more about the video. Thanks.
So the conclusion of that story is, unbeknownst to me, Howard was actually teaching me psychiatry. So for those of you who remember the Giants from those days, they had a center fielder named Chili Davis, and he was fast as could be. And I was sitting there in the ball game with a dozen schizophrenic guys and accompanying them to the bathroom in between innings sometimes. And somebody, I don't remember who they're playing, hits the ball. Chili Davis runs like a bat out of hell, and he just has a fantastic catch. He throws the ball in, and these guys are sitting next to me like this. And in between innings, the guy sitting next to me taps me, says, "Did you see Chili Davis run down that ball?" I learned about negative symptoms, so thank you, Howard. But in any event, yeah, I was here from 81 to 85. The last time I was here, I had a full head of hair, and it was all gray. It was all black, actually. But little to nothing that I know that during my second year of residency, I was at the VA, and Dr. Feinberg was my attending for 6 months. I didn't realize who this person was. He was on the verge of publishing this paper. One thing to follow up with what Dr. Mathalon said, 150-ish publications. You mentioned Van Halen. Paul Simon was here last week, right? And You Can Call Me Al. All these papers that people write, they're et al, right? There was no et al in his papers. This was his work. You know, here's a paper, and it's Erwin Feinberg, MD. and now we have 150 authors of publications. It's a very different world. But in any event, I was just a punk second-year resident trying to learn psychiatry, and the years went on. The one quick story I'll tell you, I really owe my career to Dr. Feinberg, because in my fourth year of my residency, for a variety of reasons, I decided I wanted to move up to Seattle. I said, do you know anybody up there? And it should have given me a little hint as to who he was. I hadn't had contact with him for much of the prior year or two, and he said, yeah, I go to meetings and I sit in the back, and I'm going to paraphrase what he said. He said that Dave Dunner and I sit in the back of the room and we do some critical thinking about the people who are presenting. And so he said, I'll give Dave Dunner a call if any of you know Dr. Dunner. I didn't. And Dr. Dunner is an important guy who many decades ago defined the entity of rapid cycling bipolar disorder. So Dr. Feinberg calls me back the next day. He says, here's Dunner's phone number. Give him a call. He's got a job for you. Talking about. So I called up. This guy gets on the phone right away. He says, Dr. Cohen, he says, if you're good enough for Feinberg, you're good enough for me. Come on up and we'll meet and we'll put together something for you. And that was my career. So the next few decades go on, and Howard and I are off in the Wallowas in Eastern Oregon doing some horse camping. This was two and a half years ago. So coming up to 3 years ago, something like that, and 2 years ago. The paper had been published 6 months earlier that you saw up here by Mccarl and Stevens. You're going to learn more about it. And Howard says to me, whatever happened to Feinberg? So I don't know. I'm not sure he's alive. And so I did some hunting, and I found Dr. Feinberg, and which was just, this was such a blessing. And in the fall of a year and a half ago, Howard and I met in the Bay Area, and we spent a couple days in a friend of Dr. Feinberg's in the backyard of that friend, talking to him about his career and his work and his life. And how'd you come to this? From sleep to psychoanalytic thinking to being with Piaget and getting thrown out of Europe, and now pruning, and where does that come from? So we wanted to find out, and from those discussions we were essentially introduced and went to go meet then with Tom Insel and Rob Malenka and Mccarl and Stevens. The story unfolds, and you're going to see it in this video here. So thanks for having us. Thanks for having Dr. Feinberg. We'll show you this video. It's kind of self-explanatory at this point, and then you're going to hear from the wizard. So here we go, Howard.
Science, to some degree, is truth by consensus. In psychiatry, like many other disciplines, what happens is there's a community of academics and investigators who sometimes for legitimate reasons, because they're smart and they're insightful, and sometimes for political reasons, become influential, become the more dominant intellectual forces in that field. And because antipsychotic agents like Haldol are dopamine receptor antagonists, and they seem to reduce the positive symptoms, therefore schizophrenia is caused by too much dopamine. And it's a perfectly fine hypothesis, except it's just grossly and kind of intuitively, obviously, too simple.
But in 1982, I mean, everything, as we said yesterday, was dopamine, and pharmacologically derived hypotheses from dopamine, and the idea of brain connections was not at all in the air, even in terms of the thinking about schizophrenia. The dopamine blockade has its therapeutic effect is that there's too much dopamine. But as I said then, and I say now, you can no more be convinced of that inference than you could that the cause of low-blood pneumonia is an insufficient amount of penicillin.
One of the charming things about the Middle Ages was that there was a synthetic theory of all disease, which held that the body is a bag of fluids, and disease arises when those fluids drift out of their natural proportions. If you have too much bile, you have some kinds of illness. If you have too much blood, you're sanguine. And it was a theory that we're all just a bag of fluids, and illness arises when those drift out of their natural ratios. Well, in a lot of ways, that's what these 20th century theories about neurotransmitters were. They said the brain is essentially a bag of neurotransmitters, and illness arises when those transmitters drift out of their natural proportions. And it is the kind of theory that I think is really just a placeholder till you get to better theories.
Doctors Beth Stevens and Steve Mccarl, two brilliant Harvard neuroscientists, wrote an article as senior authors that was published in Nature on February 11, 2016. The article, entitled Schizophrenia Risk from Complex Variation of Complement Component 4, argued for and provided evidence of a better theory.
I'm Beth Stevens. I'm Associate Professor here at Boston Children's Hospital, Harvard Med School, and also at the Broad Institute at Stanley Center. My lab studies the development of synapses and how the brain wires up and how this wiring can go awry in a variety of neurodevelopmental, neuropsychiatric, and other diseases. And as a postdoc, we quite unexpectedly, identified a role for these immune molecules called complement in normal synaptic pruning, at least in a mouse, using mouse models. Complement expressed by healthy neurons and glial cells normally during development. And one of its jobs, we've discovered, is to prune synapses, these extra connections that happen during development. Now, at the time, this was particularly surprising because we really didn't think about immune molecules in the brain, in the healthy brain. Much of what we knew about immune molecules and immune signaling was in the context of disease or inflammation, or if you breach the blood-brain barrier. And we really had quite a lot to do to try to better understand how it was that this group of secreted immune molecules, traditionally associated with innate immune system, clearing pathogens, for example, in the immune system, how it was that these molecules could be doing something as precise as pruning synapses.
These findings were published in Cell in 2007. Dr. Stevens was the first author of the landmark article entitled The Classical Complement Cascade Mediates CNS Synapse Elimination. In the summary, she wrote, "These findings support a model in which unwanted synapses are tagged by complement for elimination and suggest the complement-mediated synapse elimination may become aberrantly reactivated in neurodegenerative disease."
The idea being, if you don't prune properly during development, either too much pruning or too little pruning, what would the consequences for that be on brain wiring? Could some of them... Then came Steve Mccarl. We got to know each other. We're colleagues here across the street. Our labs, we didn't even realize necessarily what each other's labs were doing at the time. But his work in his lab and at the Broad was meanwhile uncovering this amazing link and a discovery that complement C4 was a potential risk gene in schizophrenia.
I'm Steve Mccarl. I'm a human geneticist and molecular biologist at Harvard Medical School on the Broad Institute. A few years ago, a graduate student in my lab set out to understand the human genome's largest influence on risk of schizophrenia at a population level, which seemed to be arising from the major histocompatibility complex region of the genome, which has many immune and inflammatory genes. But it had been a mystery for almost a decade what specific gene in that region was causing this large genetic signal. we really try to use the genome to search in an agnostic, unbiased way for the molecular influences on disease. And what we found over the course of several years of work was that the complement component 4 gene, that many different alleles or variants of that gene seem to explain a very large amount of this large genetic signal. And so the human genome led us to complement component 4, and then we were aware of this recent work from Beth Stevens' lab, which had shown that complement proteins could regulate the illumination of synapses by microglia at key times in the development of the brain. And we collaborated with Beth for several years to show that complement component 4, C4, can actually regulate this process.
Nature published Stevens and Mccarl's paper. The New York Times picked up the story in a front-page article. The author wrote, "People who carry genes that intensify or accelerate pruning are at higher risk for developing schizophrenia." David Goldstein, a professor of genetics at Columbia, commented, "They did a phenomenal job. This paper gives a foothold, something we can work on, and that is what we've been looking for now for a long, long time."
So my name is Seth Cohen, and I'm a psychiatrist now at Atascadero State Hospital in California. Way back in 1982-ish, I was a second-year resident at the VA in San Francisco at UCSF, and Dr. Erwin Feinberg was my attending. He was a fantastic mentor, and I had some knowledge of the fact that he had proposed a hypothesis having to do with schizophrenia and aberrant pruning, but not much more than that because I was a fledgling resident. So if we scooch the clock ahead, a couple years ago, there I was early one morning having my coffee before going off to work and reading the New York Times, and there on the front page is this article describing how some Harvard neuroscientists had come up with a discovery noting the complement C4 protein and pruning and schizophrenia, and I thought, oh my goodness, this had something to do 35 years ago with Dr. Feinberg. And but in the course of my busy life, I kind of let things fall by the wayside and didn't give it much more consideration than that. That summer, Dr. Rosenbaum and I were off backpacking in the Wallowas in Eastern Oregon, and Howard says to me, "Seth," he says, "you know, what about that article in Nature, and what about pruning, and whatever happened to Feinberg?" So one thing led to the next, and a year ago in the fall, I was able to track down Dr. Feinberg after not having had any contact for 35 years or so, and we arranged to meet.
Dr. Feinberg is Professor Emeritus of Psychiatry at UC Davis. In the mid-1950s, after completing a psychiatric residency at Mass Mental Health, Dr. Feinberg went to NIMH to study hallucinations. However, he became interested in sleep and left as a sleep researcher.
Very early, in about 68, we did a cross-sectional study of sleep in 5- and 10-year-old children, and then early adolescents, young adults. And we observed at that time that there was a huge change in the amount of deep sleep over adolescence. This was grossly apparent in cross-sectional studies, and the changes of the order of 50 to 60% between age 10 and age 20, say. And it nagged at me ever since I made that observation as to what caused it. There is no brain condition compatible with life that changes the sleep EEG as much as normal aging or normal maturation does between age 10 and age 20. And so it must be important. I had no idea what might be causing that, but it was always nagging at my mind that it was very important. And you could see this tremendous growth in the connectivity of the brain in the first years of life. But you couldn't see the change across adolescence, and that was not known until Peter Huttenlocher did his study, because it requires electron microscopy to see these synapses and to measure these synapses.
Dr. Peter Huttenlocher was a pediatric neurologist at the University of Chicago. Employing an electron microscope, his research focused on staining and counting billions of synapses. His stunning paper in 1978 documented a decrease of synapses during adolescence and introduced the concept of pruning. The N was small, the skeptics were many. However, Dr. Feinberg embraced the article.
That was a big aha moment for me when Huttenlocher published this. So, and I knew I might have ignored that paper because, frankly, there was so much crap being published in neuroscience, and that's even more true now. But I knew that Huttenlocher was a meticulous researcher. So when he published that paper, I paid strict attention. Moreover, it explained what had been nagging at me, which I had no explanation for.
Synaptic pruning during adolescence provided a mechanism to explain the decrease in deep wave sleep that occurred during adolescence, a problem Dr. Feinberg had been trying to reconcile for 20 years. In addition, Dr. Feinberg noted that researchers had observed massive change of brain metabolic rate, the emergence of adult cognitive capacity, decreased ability to recover from brain injury, decreased ability to learn a new language without an accent, and decreased functional plasticity. All of these could be explained by reduced connectivity over adolescence. Part 1 of the paper argued there's a profound reorganization of the brain during adolescence. Part 2 related this to the strategy nature uses to construct nervous systems, to overproduce neuronal elements and cut back on those that have not gotten successfully integrated.
And part 3 was, if you have such a massive change in the brain going on, it might sometimes be imperfect. And if this change is, all these changes could be understood as the result of synaptic elimination. And if there are errors in synaptic elimination, either too many or too few synapses eliminated, you could get mental illness. And I pointed out that this could provide an explanation for why the age of onset of schizophrenia is often at the end of adolescence, in the early 20s. A lot of ideas come as I'm writing the paper, and I'm not sure where they come from, but they sort of suddenly, naturally fit in with what I'm writing. And I am certain that when I started that paper, I had no idea that it would include the hypothesis on schizophrenia. But when, in the course of writing that paper, it popped out. And I was working in Seymour Kenny's lab, and I showed him this paper to get his comments. And he said, "Oh, we'd like to publish that in the Journal of Psychiatric Research." And I love Seymour Kenny. He's just a marvelous human being. And if he wanted me to do that, I would do it. But I really wanted to publish it in the archives. which has, its circulation is this compared to this much in the Journal of Psychiatric Research.
In 1981, Dr. Feinberg presented his paper at the American College of Neuropsychopharmacology, a cutting-edge meeting of neuroscientists. He describes the response.
Most totally indifferently. And there just was not any great interest in it. It seemed too far-fetched to people. I don't even know that it's fair to say that. It just didn't turn anybody on.
Between 1982 and 1990, only 5 articles cited Dr. Feinberg's paper. None of them discussed the pruning hypothesis. The good news, nobody seemed to disagree with his thoughts. The bad news, nobody seemed to be paying attention.
What I want to say is it was very disappointing. And it felt unfair to me. I thought there should at least be some interest in this, if only because the phenomenon of synaptic elimination and the fact of this strategy of overproduction followed by elimination for constructing nervous systems, if only for the fact that is so interesting. It's just really interesting, but other people weren't interested in it. And they were, everything was the dopamine hypothesis and stuff related to the dopamine hypothesis. And the dopamine hypothesis is certainly important, but that does not tell you how the organization of the nervous system gets distorted and disrupted.
My name is Mccarl Keshavan. I'm a professor of psychiatry at Harvard Medical School, and I'm also the vice chair of public psychiatry at the Beth Israel, the Deaconess Medical Center, Department of Psychiatry.
In 1994, Dr. Keshavan wrote a paper, Is Schizophrenia Due to Excessive Synaptic Pruning in the Prefrontal Cortex? The Feinberg Hypothesis Revisited. He recalls the research that led to this paper.
I began to get curious about the characteristics of sleep alterations in schizophrenia. And we did our first study, which was in the early 1990s, late 80s. We found that there was a fairly substantive reduction in delta sleep, or deep sleep, or stage 3 and stage 4 non-rapid eye movement sleep. So, and also at the same time we had just about begun our imaging studies, and we were involved in some of the early MRI studies and CT scan studies which were beginning to show loss of gray matter. And we were coming up with findings that there is delta sleep loss, there's also structural gray matter reductions, and there was also a reduction in the prefrontal synthesis of new membranes. So we were looking for a conceptual model, how to connect all of these findings together. So what we argued, as an extension of Erwin Feinberg's idea, that whatever normally happens during adolescence appears to be exaggerated in schizophrenia. So basically, gray matter reductions, delta sleep reductions, and prefrontal metabolic rate reductions, which all normally happen during adolescence. With a slope like that, the slope might be exaggerated in schizophrenia. The reason why they would not buy an idea of like a synaptic pruning idea is because no one had at that point shown in any way that postmortem brains of people with schizophrenia show synapse reductions.
It was just too far ahead of its time. And now what makes something ahead of its time? And I was thinking about that in the shower this morning. And I guess the main thing that makes a scientific observation ahead of its time is that it doesn't have an existing context to fit it into.
So Bob, I think was in many ways way ahead of his time in terms of thinking about pathophysiology of major mental illnesses, and specifically schizophrenia.
There is always resistance to any new idea in science, unless they are backed up by facts. And I think Feinberg was a little bit too ahead of his time.
So Bob's such an interesting character. He's very smart, but he's like your ultimate iconoclast. He's never been a joiner.
I would call iconoclastic personality, almost, I would almost say reveling in being a rebel, probably didn't help his cause. He's just being Feinberg. He's not part of our main crowd. He's just off doing his own thing, coming up with these, interesting ideas, but, shouldn't be taken too seriously. I'm imagining. So I think it was.
But he wasn't really part of the club, and I think that led to him being, in some ways, not taken as seriously. He wasn't often cited for things that he might have originated. He was a real stickler for scholarship, so he would make sure that he would cite every paper that in any way influenced his thinking. Any reasons. One was the fact that he just wasn't as well known by many people. He didn't promote the work in the same way that others might have.
I wish he had been comfortable being more self-promoting. Because one fellow published The Loss of Gray Matter and Schizophrenia and Adolescence with these movies. Someone else published in JAMA, Archives of General Psychiatry, The Medial View with a different technique, and I think we had a third paper doing it a third way, and it was presented at ACNP and child psychiatry panels. Let me put it this way, I know no successful, well-cited researcher who doesn't publish things, I think, in my opinion, this isn't meant to be a very cynical statement, in several different forms.
Some of his best work was really sort of conceptual. He was often in science, what really matters is having the killer experiment, the experiment that demonstrates something in a way that's unequivocal.
After his freshman year at Harvard College, Rob Malenka took a year off and lived with his roommate in the San Francisco Bay Area. He knew a friend who knew a friend who knew Dr. Feinberg, so he gave Dr. Feinberg a call out of the blue.
Bob, and I said, I'd like to start working in your lab. I have no experience, but here's what I'm interested in. Here's what I've studied so far. And for some unknown reason, he said, sure, a lot. And much more importantly, I can't remember, Bob would spend time with me talking about his research, giving me papers, answering all of my perhaps stupid questions. Back then I didn't know about writing grants. I didn't know about papers and how papers are reviewed. I didn't know about papers being rejected and the harsh criticism that scientists give each other. What I saw is this intellectually energetic, creative, forward-thinking, fun guy.
Tom Insel was a resident in psychiatry at UCSF in the late 1970s. Dr. Feinberg encouraged him to apply to NIMH. In 2002, Dr. Insel was appointed director of NIMH and served until 2015.
We met at the very beginning of my residency, and I was immediately attracted to this kind of a maverick kind of character who, I'm not sure anybody at Langley Porter or in the Department of Psychiatry was even quite aware of what he was up to. So he was, for me, a formative figure. He was the first person I met in my training who really turned me on to the idea that you could learn something important through science that there was an aspect of psychiatry outside of psychoanalysis that had a real future to it. Our sons were born almost at the same time, even though he was, 20 years older than I was. It was his first and my first. And I think the really memorable Sunday brunch together where we were just loading up the bagels in his kitchen, and the boys were playing upstairs, and we noticed it was raining, which was not outside, but inside. Water was seeping through the ceiling in the kitchen. And I was, of course, panicked, and in horror I ran up to see how these 2-year-olds had plugged up the sink and turned on the water full blast. But Bob, this is like classic Feinberg. He thought it was hilarious. He, in a way that only Bob could, was kind of curious about, gosh, how did these guys figure out at age 2 to plug the sink and to turn on the water full blast? They must have really schemed well to do that. So
Seth Cohen reestablished his connection to his mentor.
One of them was, although you were ultimately responsible for the patient care, you asked if I would please do the physical exam, because if you tried to use the stethoscope, you're not sure you would put the right part of it into your ear. I was happy to oblige that. You having said to me, and I can hear elements of it as we've had these discussions. You said, Seth, if ever you begin to get to the point where you feel like you know something, that's when you're in trouble.
And Drs. Feinberg, Stevens, and Mccarl all formed a new connection.
When my student Ashwin found his 1982 paper at the library, he practically came running back from the library, and he said, Steve, you've got to read this. And we sat down and read it together, and both had this reaction of, holy shit, who was this guy? He had thought about so much of what we had kind of come to be thinking about through this belabored process around genes and alleles and molecules. This Dr. Feinberg's really clear thinking had anticipated, really clear thinking from observing patients, from comparing changes in their symptoms to changes in their sleep patterns and in EEG recordings and things like that. And he had anticipated a lot of the things that we had come to be thinking about through this other process, and it was like finding this kindred spirit. Mm-hmm. We were amazed at the extent to which he had been thinking about this problem in a way that was very different from how his contemporaries were thinking about it, but actually quite similar to how we had come to think about it through these genetic and molecular studies.
In June of 2017, 35 years after publishing his paper, Dr. Feinberg flew to Boston to meet with Dr. Stevens and Dr. Mccarl. On a lovely Saturday morning, Steve Mccarl graciously hosted a brunch at his Cambridge home. 3 scientists sat down to meet, to talk, and to celebrate.
Hi there.
So nice to
It's
see
nice
you.
to see you.
Hello. Nice to meet you finally. I'm so excited.
It's really wonderful work you've done.
Thank you. You too. I'm really
You
grateful.
know who he is, I presume.
Absolutely.
I don't have to make a formal introduction.
I'm really grateful to your colleagues for connecting us.
I am too.
Oh my God, it's so cute. I love it. Wonderful.
Okay. Well, to say this is gratifying or flattering or whatever would be a huge understatement. And I am so grateful to Seth and Howard for appearing suddenly at this late stage of my life, and so grateful to Dan and Judy for setting up this presentation. And it feels very good to be back at UCSF, where I had many happy years and some not so happy. And this is part of, it's been broken up into several rooms, but this is where I gave my first lectures in the newly reorganized Introduction to Clinical Psychiatry that I established with a group of my colleagues, some of whom are here today. And that was a fun course. We introduced small group teaching and patient examination and things like that. What had been a more academic or ideological course. Anyway, it feels extremely good to be here, and I'm very grateful to everyone for all that they have done. A little bit more, and I would get maudlin, but I just want to review some of what has already been mentioned in the course of the video. And one thing that has always befuddled me is that even in good, strong, rigorous science, there is much greater weight given to a small biological effect if it's been measured with a huge machine than to a huge biological effect if it's been measured with a small machine. And I was introduced to this paradox very early in my research with the example of brain changes in adolescence. And so there is this massive decline in slow-wave EEG in non-REM across adolescence, and that's been known since before 1968. This was one of our very early studies, and each of those data points, these X's represent small groups, 5 or 10 subjects. But you see there's this huge change between childhood and age 20, where you go from 400 units to under 200 units. So we were interested in the long-term delta EEG trajectory of this phenomenon, but we only had ink-written EEG records, which is all night EEGs are several miles long. And I had data from age 5 to 95 years, and Arthur Parmalee, a pediatric neurologist at UCLA, had data from infants from a little under 1 month to 5 years. Well, it would have been enormously expensive to scan and digitize those miles of EEG record. So what we decided to do was to use sampling and hand measurement of the wave amplitudes. And Satoshi Hibi, who you may meet, he's going to be coming sometime today, made these measurements at the San Francisco VA, and then he went down, he took all his equipment and went down to Arthur Parmalee's lab and made the measurements there. And here's the equipment he packed. And using that clever device, he produced the following curve, which is a crude curve, but it does show there's enormous increase in infancy and there's enormous decrease in adolescence. And when we published these data in 1977, we said, well, this rapid growth in delta amplitude during the first years of life may be an electrophysiological correlate of the increased connections in the cortex that occur with postnatal maturation. And I cited Connell's work, and a little sidebar to Connell's work. His work was basically not really known or discussed. And Connell was a man at Harvard who was a pathologist. And each year he would do an atlas of the human brain at different ages, and he published the atlas. And at the time I was writing this paper, the medical library here was on the 4th floor of this building. And Connell's atlases were kept in a locked case behind glass, and I had to sign them out each time I wanted to examine them. Now I find to my dismay that they're just available on the shelves like any other books. But anyway, they were very special at that time, but nobody, they were so well protected that nobody looked at them. At any rate, so Connell's data could explain that phenomenon, but the explanation did not come until Peter Huttenlocher published his data on synaptic density in the cortex. And Peter did this work by himself, and he was a very busy clinical neurologist, pediatric neurologist, for his entire career after the NIH, and he did these studies, and that's what he came up with, and he followed up on this also. So then I published the paper that's already been noted, and there were 3 arguments in this paper. The first argument was that the human brain undergoes a massive reorganization in adolescence. Well, this is now commonplace knowledge. Every checkout person at Safeway knows this. But at that time, adolescence was a terra incognita in terms of neuroscience. And yet the evidence was already there. There was this massive decline in deep sleep. There was a decline in cerebral metabolic rate, which we traced in the literature, and then it was later documented very powerfully with PET scans. But there was just scattered data, but they pointed to exactly this phenomenon. And then there's decreasing anatomic plasticity, which is the ability, say, to recover from a lesion, a brain lesion that causes aphasia. Well, I did very thorough research on the literature on this question. As far as I know, the data are still very limited on this very important question. And at the time that I was doing this literature research, the estimates of the lesions were not nearly as accurate as they could be today with MRI and so on. They were based on ventriculography and so on. So that's still, I think, a very interesting area for research, though it may not be sexy enough for the study sections. And then there's the emergence of what I called functional plasticity, which is the ability to solve problems, to set up temporarily in your brain a neural structure which, when it is run, can produce a response, and that is used over again. And I pointed out that setting up such a neural structure may require having fewer choice points and fewer misdirections, and so that decreasing synaptic density could account for both the loss of anatomic plasticity and the emergence of functional plasticity. Well, it's a hypothesis still, but it is, I think, a viable one so far. And then this was the second line of the argument. The first was that there were all these changes taking place in adolescence. The second was that they could all be explained by synaptic elimination. And the third was that if you have something so complex going on over a prolonged period, it could sometimes go wrong. And then we were able to do a statistical analysis and a curve fitting analysis, and to show that the curves for synaptic density and delta wave amplitude and cortical metabolic rate were statistically similar, as well as visually similar, and they're measured in such vastly different ways. Electron microscopy for synaptic density, delta wave amplitude with a millimeter ruler, and cortical metabolic rate with PET scans. So that encouraged us, or encouraged our thinking. And it is, I think, if one looks at it historically, that the pruning hypothesis, which had been ignored from 1983 to the early 90s, really only became of interest when cortical thickness was measured with MRI machines, and people were able to show that the cortical thickness was diminishing in adolescence. Well, look at the relative magnitude of the changes in cortical thickness and the delta EG. There's about a 10 to 15% decline in cortical thickness over adolescence. There's a 65 to 70% decline in delta wave amplitude over adolescence. And where I was brought up, there was more interest in a big effect than in a small effect, but that is not the case if the big effect is measured with a small machine, and vice versa. So don't be seduced by technology is my main cry. So we've continued to focus on sleep EEG, and with Ian Campbell in my lab, we have made really nice longitudinal measurements of that change from age 6 to age 20 and delta wave power. Now we're measuring power. delta wave, not with a millimeter ruler, but with an FFT analysis of EEG. This is what the old cross-sectional curve based on fewer subjects looked like. This is what the longitudinal curve looks like, and I think it's really quite a fascinating curve, and this is the only such data that exists at this day. So we had one group, one group of subjects starting at age 6 and going to age 10, those are the white circles. Another group starting at age 9 and going, these are the green triangles, going to around age 15. And then a third group starting at age 12 and going to age 18. And notice how amazingly these groups meet each other. These are independent longitudinal groups, and now the ends are quite respectable, at least 30 in each group. And the other thing to notice is instead of the steep decline we saw with the cross-sectional data, the delta power stays pretty high until about age 12, 12 and a half. And then it plummets to about age 16 and a half. And so I think that furnishes, as I discussed with Beth Stevens, some clues as to where they should look for the changes in synaptic elimination due to the microglial systems that they discovered. That is, it would be interesting to look right around here, where the curve is just about to begin its decline, and here where it's changing rapidly, and then here where it starts to flatten out. So that might ultimately lead to further connection between the basic neuroscience research and the clinical human research that we've been doing for so many years. So I think I'd better skip along now. I was going to mention a few other thoughts I've had about schizophrenia, which are not much discussed these days, but which I think still are puzzling. And one of them is why brain injury never causes schizophrenia. And it never causes schizophrenia. And yet there's such myriad kinds of brain injuries. So why is it that injury or lesions? Well, there are two obvious possibilities. One is that the kind of lesion that will cause schizophrenia is incompatible with life. That is, it's in some subcortical area, and you can't survive with it. The other is that the kind of integrative defect that is schizophrenia, such as the impaired corollary discharge mechanisms that Dan and Judy work on, that this kind of subtle impairment can't be produced by attacking the brain with a hammer. And so I think it's an interesting question. Now, another thing that has bothered me is the assumption, and it's still an assumption, that early treatment of schizophrenia leads to better results. Now, that might absolutely be true, that early treatment leads to better results. But I don't know of any evidence that shows that, any rigorous evidence. And that would be a very interesting question to get an answer to, because it has a lot of implications for how we treat our patients and how early we give neuroleptics and things like that. But it's just a given in the literature that early treatment of schizophrenia gives you better results. Well, it's true in many medical conditions, early treatments does give you better results. But there are also many medical conditions where it doesn't, and where it leads to very unfortunate kinds of interventions. So that's another thing that is on my mind. So now I'm finally wrapping up over 60 years of psychiatric and neuroscience research, and research labs work differently, I think. In my lab, the quality of our research really reflects the tremendous contributions of my research associates. Many of them are much smarter than I am. I'm not saying that out of modesty, it's just a fact. They have had such devotion to getting the answers right, to correcting my errors, to seeking the truth. It has been just an amazing experience. And I can't think each of them. There have been many who've come through my lab, but I want to mention two who were with me for the longest time. Jonathan March came to my lab as a conscientious objector in 1972. He was the first in his class at Swarthmore in math and physics, and he never really left the lab. He made contributions in every area. He had an amazing ability to look at a bunch of numbers and see visual patterns in them. It's just really extraordinary. He's now a programmer, a scientific programmer in Austin. We're still in regular contact. And Ian Campbell joined my lab in 1990. He was a summa graduate from UC Davis in physiology. And over the course of time, he's become one of the country's, and in fact, one of the world's experts in human sleep. And this is Jonathan March 13 years after he came to my lab, and then more recently. And there is Ian Campbell, just about the time he joined the lab, and here he is this year. You can see that working with me has a terrible aging effect on people. So, and this is Satoshi Hibi, who may show up here, who spent World War II in a relocation camp where he and his family were moved. And then when he was released from the relocation camp, he was given the honor of being drafted into the Air Force. And after serving in the Air Force, he came and worked at UCSF for a while, worked with me for a while. But Satoshi is mainly an artist, and he went to Japan 40 years ago to teach art and stayed there for 40 years. And he came back a couple of years ago, and he's living up here in the Sequoia now. And we had a visit to the Museum of Modern Art a month or so ago, and there is Satoshi. And Satoshi said, when I invited him to come here, he said, how formally should I dress? And I said, however formally you dress, Satoshi, it will be more formal than the way I dress. So you should feel comfortable. So again, I want to thank all of the people involved for putting this together. I want to thank my friends in the audience who I see and I look forward to chatting with. And of course, I think we also owe a debt to the institutions that make our work possible. You fight with your bureaucrats all the time. On the other hand, you couldn't do the work that you do without their pulling their oars and working for you. And sometimes I know that I did not appreciate that as much as I should have. So this is a very belated thank you to them as well. So thank you.
Well, I think that we could eke out maybe one question before we hit the 9:30 mark and we can wrap up. Some of us will have some opportunities to talk to Dr. Feinberg and Drs. Cohen and Rosenbaum at more length after this. But does anyone have a burning question they'd like to pose to Dr. Feinberg before we wrap up the session this morning? Yes.
Sorry, given that I'm a therapist and very interested in treatment, would this have implications for treatment that you've already thought of?
The question is whether the work that you've discussed today have any implications for treatment that you have thought of.
Basically, no.
Okay. Well, since it was a quick question, I'll take one more.
Yeah, this was really great rounds. It was nice to get the historical perspective of a lot of over time we don't get to see the origins of some of the great theories that we grapple with in psychiatry. And when you came up with your theory, there weren't a lot of big, powerful machines, as you mentioned, and you came up with this pruning hypothesis. And a few, 3-4 decades later, Mccarl and Stevens were able to find some rigorous evidence for this theory using big, powerful machines and an unbiased approach. And so it's really nice to see the concept meet up with the data that was discovered through an unbiased approach. And so, some of us in the audience are young scientists, and I just wonder if you have advice for us in terms of how to approach a scientific question. Should we lean more toward the conceptual side, or should we sort of search amongst these big pools of data that are being generated with big, powerful machines?
So basically, Dr. Barkley is asking if you have advice for young scientists about giving more weight to sort of conceptual aspects of the scientific work, or to really delving into the data that are generated now by these large machines that give us very precise measurements, and where should a young scientist, now that we have so many of these kinds of machines available, be spending their time? Where should their emphasis be? Do you have any advice?
Well, I actually do have some advice, not on this specific question of theory versus empirical data, but on the question of, on the issue of how does one choose a scientific problem if one is a young psychiatrist. And in a number of medical fields, the scientific problems are very obvious, and there is a natural kind of basic science that goes with those fields. Neuroanatomy with neurophysiology with neurosurgery, for example. But in psychiatry, the basic science could be anthropology, sociology, biochemistry, genetics. So what do you choose if you want to do research that goes beyond work in clinical psychopathology and has a more rigorous base? And there is no one answer to that, but my answer in general terms would be, one, choose a problem that really interests you. Two, try to choose a problem that might have some bearing on the mind, how the mind works and how the mind gets diseased. But don't choose a problem because you think it's going to give you the answer to schizophrenia or bipolar disorder or whatever. Choose a problem that just really is intrinsically interesting to you and that might, if you're very lucky, and I was lucky, and it's nothing but luck, and I was lucky that it did have some bearing on the clinical issues.
Okay, so on that note, thank you all for coming today, and thank you to Dr. Cohen and Rosenbaum and Dr. Feinberg.